The next speaker, so Filipe Cabreiro from Imperial College London in the UK, and he is going to be speaking on a topic that is very dear to all of our hearts and all of our guts, tweaking microbial metabolism with drugs to improve aging.
All right, thank you very much, Alex. Thank you, Alex, again for the invitation. Also, Morten, it's a pleasure to talk at this very exciting conference. So yes, our lab is interested in host microbe interactions, in particular how microbes can regulate or modulate aging. So let's see if I can change. Oh yeah. So the question regarding whether the microbiome can actually have an impact on aging is quite an old one, and it all started, as far as I can tell, from the work from Metformin. It was theoretical work where he strongly believed, and I emphasize the belief in this, that the gut microbes could play a key role. He even wrote an essay on the topic, which we called The Prolongation of Life, and I love the subtitle, what it really says it all, optimistic study. So he didn't have any evidence to support this, but he was a strong believer in it, to the fact that he drank fermented milk throughout his whole life. But it took quite a long time to get to the point where we do have actually evidence now that strongly links the role of the microbiome to aging. And even Alex's great work, we can even now actually look and use the microbiome to predict human aging, which is a remarkable find.
Philip, sorry.
Yeah?
Are you sharing your screen? We can't see your slides. So we just want to make sure if you are.
Yeah, I'm sharing it, yeah.
Yeah, sorry, I thought that I should have interrupted before, but...
No, you cannot see my slides changing?
We cannot see your slides at all right now, so we're wondering if...
Let's ask the operator of the Zoom channel to see, because they have changed the system.
We have it here, so we cannot see your slides. So if are you sharing your screen?
Okay, can you see it now?
Perfect.
Perfect.
Yep, perfect.
All right, sorry about this.
So just on Metformin, perfect.
Yeah,
Thank you.
Yeah. All right, so
great,
apologies
fantastic.
for this. So indeed, Ali Metformin was one of the proponents that indeed the microbiome played a key role, and he wrote this very interesting book about this particular role. But it was really, it took quite a long time indeed to get to the point where evidence existed to strongly and causally link the role of the microbiome with aging. We work from Alex himself shows that we can predict human aging looking at the microbiome, and work from many of our colleagues are now causally linking really the role of specific microbes and metabolites from these microbes to most of the hallmarks of aging. In this review that we wrote last year, we even go further to suggest that actually the dysbiotic environment within the gut that can lead to changes in the composition of the microbial species can actually or potentially be considered one of the hallmarks of aging. And of course, this is a very complex ecosystem, modified and modulated by many factors, including diet and drugs. And this is really one of the aspects of the work that we do. And we'll talk in particular one of our pet drugs, which is Metformin. So Metformin is, as many people know, a putative DR mimetic, and it's been now actually widely used in model organisms to test whether really it has a positive impact in modulating lifespan. Work in worms from many groups really shows that this is a very robust effect. You can get a very strong effect on lifespan, but also health span. Work in insects, including recently work in flies, also shows that this is an evolutionarily conserved effect. And of course, the work by Rafael de Cabo and Anisimov in Russia also really seem to make a very strong point that indeed this may be a potential therapy to improve aging. But as Nir Barzilai mentioned in this response to his questions, what is really striking is the data coming from humans. So this is the data from a UKPDS study where they looked at type 2 diabetic patients on Metformin compared to matched controls with either other drugs or even healthy patients. And you can see that indeed Metformin monotherapy increases survival in patients even compared to human healthy individuals. And I think that is really quite a striking phenomenon. But was quite, so of course, there's a lot of mechanisms by which metformin impacts on a host physiology. But one of the, what we think could have been potentially an important discovery was made in around 2013, work led by us and also by other scientists in that same year in different organisms, including C. elegans, mice, or humans, are now actually showing that the role of metformin by regulating the microbiome and the microbiome function could impact on host physiology. We were the first ones to show that this was essential for the longevity effects that we observe in C. elegans. And in the later years, in 2019 and 2020, we now show specifically how this works and how it also improves health span. But what is really intriguing is this link. When you look at how, or potentially how metformin works, it seems to be linked to the effects of E. coli itself. And in type 2 diabetic patients, for example, there's a strong dysbiotic change in the microbiome. And what metformin seems to be doing is to normalize those effects. And in particular, it seems to lead to an abundance of Escherichia coli to produce these positive effects. But the link between metformin, E. coli, and a dietary role in these phenotypes was very unclear. And this is really where we took the past study from. We try to understand how diet could influence the effects of metformin. And this can be done very nicely with C. elegans. Normally in the C. elegans context, you would have worms on a specific type of nutritional content where you put your worms, and you can see then an increasing dose of metformin, you see this bell-shaped curve in longevity. But now when you start changing the nutritional content of that same media, you can see now that this curve completely changes its behavior. In a very, very rich media, you can see actually that metformin only seems to have a very small effect on longevity at very high concentrations. And another key finding that we have already done in 2013 was that, in fact, if you used bacteria that are resistant to the effects of metformin, metformin no longer extended lifespan at any concentrations tested. In fact, it only seemed to have a toxic effect. So this really supported two things. One is that indeed the effects of metformin acting through bacteria seem to be very important for longevity. And the second one, that these effects were strongly regulated by diet. So diet is very complex. We tend to simplify it based on macronutrients often. And the FDA alone only focuses on the role of about 100 metabolites or nutrient components. But the reality is each one of the foods that we eat are probably composed by around 20,000 different compounds. So how do we go to study now that level of complexity? And this is something that we try to do. We developed a technology where we started studying how 400 different metabolites approximately could influence the effect of metformin on, for example, E. coli growth. What you see here is a bacterial growth curve on a control situation in red. When you treat with metformin, there's an impairment in growth. When you challenge the system now with different metabolites, you'll find that some will antagonize the effect of metformin, some will have absolutely no effect, and some will actually make metformin impair bacterial growth even more. But we wanted to see, according to our predictions, whether indeed if just the effects of bacteria could be fully predicted by looking at the effects on the host. And for that, we utilized a system where we have a transcriptional reporter line for a gene which is involving beta oxidation and mediating ultimately the effects of dietary restriction. So when you challenge this system with metformin in a sensitive strain of E. coli, these worms will glow bright green. And now we wanted to study how metabolites could challenge and change the expression of this transgene, either by shutting it down or making it even further activated. And we tested about 5,000 phenotypes in order to comprehensively study all these different interactions. So the data is very complex, and I'm not going to go into very fine detail, but what ultimately we wanted to know was whether there was indeed this, that was our hypothesis, whether there was this correlation. And we were surprised to see actually that correlation was not as strong as we thought it was going to be. We couldn't fully predict the effects on the host by looking at bacterial effects. In fact, nutrients that can rescue the effect of metformin in bacteria, here in this line, for example, some of them can have a very opposing effect on the host. And when we start looking at then how these things explain the effects on the host, this really revealed that immediately that the bacteria had an amazing ability to interpret signals from nutrition in the context of a drug and this was completely unclear how this actually worked. There was nothing that we knew that would explain this. So we started enriching these nutrients into their metabolite classes. And again, if we could explain the data in C. elegans by just looking at the effects on bacteria, this would be a mirror. The two enrichment maps would be a complete mirror, and they were not. In fact, only a very small subset of the metabolites that could rescue the effects on E. coli could also rescue the effects in C. elegans. But when you place these nutrients also into metabolite pathways that are known, for example, CAG pathways, we can do a different sort of enrichment. And what we found, looking at this similarity between the two data sets, the two things came up. One was a metabolic pathway, galactose, and the other one was the phosphorotransferase system, which is actually a signaling pathway. And this was really interesting because we would never imagine that a signaling pathway could be mediating such effects. So in order to understand the link between metabolism and transcription or signaling, what we did, we performed proteomics in bacteria treated with metformin. We did the usual thing of enriching for functional categories, and we found an enrichment for glycolysis, TCA, and arginine degradation. But then we asked the question, what were the transcription factors that were regulating those expression changes? When we performed that sort of enrichment, we found that 4 transcription factors were significantly enriched, and I'm going to focus only on CRP at the moment. So our 3-way screen had revealed, as I said, the PTS pathway, the signaling pathway, and now the proteomics data is identifying the transcription factor that literally sits at the bottom of that signaling pathway. When we start doing a lot of molecular work, both Western and other techniques, to really understand whether metformin activates and overexpresses CRP. We observe all that and we validated this. But when you start doing also genetic work and studying their effects on the host, when you abolish entirely the pathway and the ability to produce the cofactor or even the transcription factor itself, metformin no longer extends lifespan. What you see here is metformin extending lifespan, the red versus the black in full, and in the dotted lines, when you put these worms on a bacterial mutant that doesn't have any of these elements, here is just shown CRP, the effect is abolished. So really showing that CRP was a key regulator and the pathway was a key regulator of the effects of metformin on the host. But then at that point, we were super curious and we wanted to know whether we could actually start tweaking bacterial physiology, metabolism, and signaling pathways in a way that we could obtain positive effects even without the drug. So what we did, we overexpressed CRP genetically, but we created a system where we could make it dependent or independent of its cofactor CMP. And what you can see here is that if you overexpress CRP, you can make these worms live robustly much longer, but only if they can produce the cofactor that activates the transcription factor. So we found this very beautiful node between transcription and metabolites that regulate metabolism to confer and alter host physiology. So, but what this gave us too was a tool that allows us to use another different set of techniques to understand what was downstream of these processes. We had a system now where we could do metabolomics in bacteria that are sensitive to metformin. You can see a lot of metabolite changing. If you perform the same experiment in a mutant that doesn't have CRP, it's quite amazing how these cells are almost blind to the effects of metformin. And we have a system now that it can overexpress the same transcription factor, but even without metformin at different doses. When you put all this data together and triangulate the data to find commonality between metformin effects and CRP overexpression that are completely abolished on CRP, you end up with a very small subset of metabolites. When you start actually looking at what these metabolites are, this is where sometimes history teaches us a lesson. And we found that a lot of these compounds were actually being tested about 100 years ago as potential molecules for treating type 2 diabetes. And this was a very humbling thing, actually. So we started studying those in all detail. We ruled out all the molecules we had found, and we found that actually arginine metabolism in bacteria seem to be a key player for the effects of metformin. Arginine catabolism has two pathways. One is called the AST pathway, and the other one is the putrescine metabolism pathway. And we can play almost a plug and play situation where we can block one pathway or block another one. And when you block, for example, the AST pathway, we see an activation of this dietary restriction reporter that we had used, that had identified the mechanism for metformin. When you block SPB, which leads to an accumulation of agmatine, you see a further increase in the transcriptional reporter. If you block both, it becomes even more robust. And if you create a quadruple mutant that can no longer activate agmatine, you completely suppress the activation of this dietary restriction reporter. And this were all translated into lifespan effects. And we wrote, and so therefore we concluded that agmatine could be a key player in mediating dietary restriction. So we went even further. We supplemented agmatine both in C. elegans and in flies in collaboration with Alan Koshme at the LMS, and we could see that agmatine alone could also extend lifespan robustly. What about metformin and agmatine in humans? We did, in collaboration with Christoph Coletta in University of Kiel, we did a metabolic flux approach, a metabolic modeling approach, where by knowing the bacterial species that exist in the gut and each one of their metabolic maps, by having the information about diet, we can actually start predicting whether a specific community of microbes can actually produce a molecule of interest. So we apply this to our system and I start asking the question, first, what are the key microbes that are producers of agmatine? And in several cohorts, a new cohort that we have access to, and 3 previously published cohorts, E. coli came as one of the most, if not the most important species producing agmatine. We even tested whether indeed patients, type 2 diabetic patients treated with metformin have an increased ability to produce agmatine, and we found that this was significantly increased. And this was also validated in a longitudinal data set. And finally, we start asking also questions, what other molecules are produced when the bacterial cells need to produce agmatine? And we found that there was a strong increase in the production of short-chain fatty acids, which are known to be important molecules for regulating health. And we also found that they produced gases that are known to be one of the negative or undesirable side effects of metformin uptake. So just to summarize, basically we developed a new high-throughput screening approach that revealed new processes in bacteria that could have not been found in any other way. Something quite remarkable, that we have a signaling pathway, almost like the FOXO pathway in host cells, that is mediating the effects of nutrients and drugs, and by being activated can change metabolism to produce molecules of interest. And we also found that these effects are conserved across taxa and may potentially explain even the longevity effects in humans. So I'm going to shift very quickly to a second story. We got interested also in how we can measure health and how interventions can improve health. And this was a beautiful work from Clara, a postdoc in my lab. And she came with this technique, atomic force microscope, where by using it, we have a resolution that is actually below scanning electron microscope. And this really gives us an opportunity to look at certain processes that cannot be done with higher or less resolution microscopes. So what we can obtain by using atomic force microscopy is this a very fine idea of roughness on the surface, and also a measure of what we could call stiffness, where by applying a specific force, the tissue will rebound, and that will give you an idea of the characteristics, biophysical characteristics of that same tissue. We looked at worms at different ages, and what we observed is indeed, as you apply different forces in worms of different ages, as they get older, they have they become much less stiff, and they take longer to return to their initial state. We can apply a formula called the Young's modulus, which converts mathematically these force curves into something a bit easier to interpret, and this is exactly what you see, that as worms age, the Young's modulus is actually decreased. We also can obtain images, topographical images of the skin or the cuticle, let's put it in more correct terms, and we can see that there was indeed a degradation of the cuticle, and this can also be quantified and here is exactly what you see, and it's almost quite stable to midlife, and then in very old worms you could see a very clear decay with aging. We can then start testing genetics and dietary interventions, and we've done all that. So if you're interested more about this, you can read the paper. But I want to just focus on two things here. Is we start playing with starting genetically modifying bacteria, and you can see that some of these interventions can robustly extend lifespan. So bacteria that don't have these genes in the Krebs cycle, they increase lifespan quite robustly, and they also, with aging, which is the Young's model is very low in old worms, but this seems to be improved, significantly improved when worms are grown on these two types of bacteria. And this is at chronological age, where you can see this is a chronological age, and this is at physiological age. Also with metformin, as I said before, we can see something. We can see that indeed with aging, the Young's modulus decreases. The stiffness becomes worse as the worms get older, and metformin is capable of maintaining that stiffness and that the quality of the tissues for a very, very long time, only decaying very slightly in the very, very late life. We also showed in the manuscript that these effects were strongly dependent on the bacterial type, as if you grow worms on bacteria that develop resistance to metformin, you no longer see this positive effect on stiffness that is observed with metformin treatment with aging. So I'd like just to finish really to thanking everyone who previous and current lab members have been great in producing this work, our funders, our amazing collaborators, and I'll just use the opportunity that the lab will be expanding and we'll be recruiting new PhD, research tech, and postdocs. So if you want to know about those opportunities, either contact me directly on my email or check on our website or on Twitter. Thank you very much.
Thank you very much, Filipe. Great talk. Again, microbiome plays a huge role in aging. Thank you very much for the work you are doing. We have a lot of questions, as you can imagine, and it would be great if you can go on Slack and later on answer them. I'll try to be very brief. So many diabetic patients have intestinal discomfort on metformin. Any thoughts on why certain individuals have this problem, and could this be an issue with the microbiome? That's the question from Matthew Janssen.
So there's two views. There's the old view that it's actually caused by direct effects on the cells lining the gut, and an accumulation of acetate and lactate, which is still very unclear. This comes from some old papers, but the most recent view is really the direct impact, which is now very strongly supported by the metabolic modeling and also by other measurements, that indeed increases in production of CO2, hydrogen sulfide by certain microbes by the effect of metformin on those bacterial cells is really what's leading to this very known discomfort of metformin uptake. So, or it could be very likely it's probably a combination of both things, but yeah, I would definitely say that the effect on the microbiome is a key driver of that.
And how can the results in worm be extrapolated to metformin effects in humans? For the question from David Garcia.
Yeah, it's a very good point. We've seen that definitely there's an evolutionary conservation of the effect. We tested this also in flies, and it seems to be conserved. I think if we're right about this, it's not been tested, and it definitely should be tested in the future. If we're right about the production of agmatine as a key intermediate between metformin, the microbiome, and the host, we've seen that by the metabolic modeling, we can definitely see that the molecule is predicted to be increased in type 2 diabetic patients with the treatment. And there's also evidence from other animal models where the supplementation of agmatine alone seems to be having very similar effects to metformin itself. So it could be that metformin acts in a very interesting way by forcing the microbes to produce these other molecules which seem to be having equally anti-diabetic effects alone. And agmatine is quite an interesting molecule too, because it follows the description of a neurotransmitter according to Enridale. So it could be some weird kind of gut-brain connection too, though it has been completely unexplored. But there's many different ways that I think that could be acting here.
Thank you for this comprehensive answer. Just on a side note for everybody in the audience, if you want to take a break, currently we do have it scheduled for 20 minutes. So any kind of bio break, or if you want to do some exercise, or if you want to continue on this note, please do so. Maybe you can do exercise and continue at the same time. So we're going to start with Anais Franco Romero at 10:20 New York, but we are going to continue for another few minutes with questions, if nobody minds, because again, lots of questions for Filipe, including for myself. Question from Alexei Strigin. Filipe, great talk and great research. Do you think probiotics enhancing efficacy of metformin might be developed?
I think so. I tend to call them more kind of synthetically modified microbes than probiotics, right? I think a very clever design of bugs to perform that function, I strongly believe that could be a way forward. We actually have data already in the lab that supports that using different types of strains and modifying certain mechanisms can actually improve dramatically the effect of the drug, or can actually suppress dramatically the effect of the drug. So the answer is yes.
Yeah, and a question from Morton. Is metformin able to inhibit complex 1 in mitochondria from C. elegans?
That's the good old one. I would say yes. We have new evidence that really seems to support that. Somehow, no, it's just the nature of the molecule, right? We've tried many different things to really have that kind of key evidence that says, yes, it's there, it's binding. And there's really nice work that seems to show that. But the definite proof isn't there yet. No one has ever been able to really make that key piece of evidence. But a lot of the evidence points to that, including new data that we have. So yes.
So a question from myself then. I mean, you mentioned our work on microbiomic aging clocks that we developed a while ago and published recently with Vadim Gladyshev, who presented yesterday. I wonder if you have taken a look at the composition of those of the microbiome and the species that quote unquote make you look younger, kind of geropositive, and species that make you look older.
I think it's a great study, and I follow that from when you submitted that in bioRxiv. I thought it was remarkable. And I think that's, if this proves to be right, what we don't know is it correlational, is it causational, right? That's where things are still a bit tricky, right? But if they happen to be causational, right, and they do regulate, they have an active role in regulating the process, I think finding therapies and ways to target those microbes that are, promoting aging, then I think it's definitely a strategy for the future, yes.
And if you've looked at some of those species, do you have any idea how to manipulate them with drugs to make your gut look
not
younger?
Yet. Not yet. When I do, I'll let you know, but not yet, no.
Yeah, would love to collaborate. If you have any microbiomes after treatment, would love to run them through the clocks. Now we have much better, more advanced clocks as well.
Okay, cool, brilliant. We'll be in touch then.
Yeah, thank you so much. So for every